Hello students, let us understand megasporogenesis today. Before we go into megasporogenesis, I hope you watched the video on microsporogenesis. We'll just recap it before we go ahead. So microsporogenesis is the production of a microspore which is happening inside microsporangium from PMC cells or pollen mother cells. And this microspore is what becomes the pollen grain. Inside the pollen grain will be the male gamete. Therefore, we call pollen grain to be male gametophyte. I hope that is clear. If not, go back please and watch the previous video. Alright, let's understand megasporogenesis today. Before we go in depth, let's have a quick summary of what the process is. Megasporogenesis, we get production of megaspore from, similar to PMC here, is megaspore mother cell, MMC, megaspore mother cell. Again, this is present inside a megasporangium called as ovule, and from there you get the gametophyte. Which gametophyte? Female gametophyte, that is the embryo sac. Therefore, inside this gametophyte will be the gamete, which is egg. So this is a quick recap. Let's understand the whole process in detail. Let's have a look at all the pictures. It's much easier with the pictures than not. Megasporogenesis. So when we learned about microsporogenesis, I hope you remember, we started with the anther and then filament. And then inside the anther we went, cut open the anther, saw that there are four different microsporangium inside, all that. Similar here is the female reproductive part of the flower, that is the carpel. Which one is the carpel? So we always say the female reproductive part is the gynoecium, isn't it? Gynoecium. And if more than one is there, we call that as the entire structure we call as gynoecium in single units. Like so, this is one single unit. This one, ovary, style, and stigma. This is one single unit. Is called as carpel. Now, if there are only one carpel, we call it as monocarpellary. Let's say there are more, like here, many, then we call this multicarpellary, and this multi can be either joined together or separately staying and all that. It is given in the textbook. Please go ahead and read it. You don't need so much of an explanation for that. So please have a look at the gynoecium or the carpel here, the single unit of the gynoecium called as a carpel. What all are the parts? We can see there is a ovary, there is a style, and there is a stigma. You know, stigma is a landing place. This part is the landing place of the pollen. Style is just a part that connects this two, and ovary is the hero right now. Ovary, when you look at this bulged part, the ovary, we need to go inside and see what is inside. So it looks like so. This is the cross section of an ovary. When you take a cross section, you see that there is a tissue like so in the center. It may not be always in the center. This is a type of ovary in which it is in the center. We will not go in depth about that. Right now, we'll understand that it is in the center, and name of that tissue is placenta. This part is called as a placenta. From the placenta, you can see that there are globular structures arising like so. Right? How is it arising? It's arising first from the tiny stalk or the filament kind of structure. From there is a bulged part arising. Right? Both of them have names. So this large tissue to which all these globular structures are connected is called as a placenta. This one is the placenta, to which there is a stalk. The name of the stalk we have to learn, and from the stalk there is a globular structure. Right. So let's see what they are. So this part when we take, or the one that I drew, I am going to take this out. All right, this part, this one out, and then I'm going to just keep it this way. This is what I have taken out. All right, that part, this part, which is the stalk, is called as the funicle. The same thing is enlarged and shown here. So Ovary, first of all, we have cut open the ovary. We are seeing placenta, we are seeing the stalk, we are seeing the globular structure, and the name of it is ovule. Wait, I'll not write here. I have to write right here. Ovule. Which one is the ovule? This globular structure is called as the ovule. I've taken it out, I've kept it like this. Alright, this way. So that is what you are seeing. Now you can, I hope you can relate. Let's see. So this stalk, this part stalk is here. And we call that as funicle, the stalk of it. After the funicle comes the entire structure itself, the ovule itself, what you're seeing here. So it's not actually a globular structure now, isn't it? So it has many parts to it. Let's see the parts. What is it? There are first and foremost outer coverings. Outer covering. It's not going proper. Covering. Outer covering number 1. Outer covering number 2. Right? The same way here also. I'm drawing only on the one side. Outer covering number 1 is called as the outer integument. This one. Inner covering is called as the inner integument. I hope that is clear. Now comes another food-laden tissue. This one. This is of importance to you. And drawing is not coming proper on the board. Okay. I'll draw again. Integument number 1. This one. Integument number 2. Integument is the outer covering. Inside that is a food-laden tissue. Where is it? Here. This is of significance to you. This is called as the nucellus. Nucellus. Okay. This nucellus has few cells, and apart from all these, you should know the orientation of it. How is it oriented? That is, you can see over this side, there is a pore kind of structure. Isn't it? There is a pore kind of structure over this one. Wherever that pore is, we call that pore as micropyle, and whichever side that pore is, we call that as the micropylar pole. Micropylar pole. And the other side, opposite side, this side, we call that as challasal pole. So I hope it is clear. We looked at the stalk first. What is the name of the stalk? Name of the stalk is funicle. Then you have two coverings called as the outer and inner integument. Wherever that pore is, what pore is it? The integument is not completely covering the entire structure. There is a part where the integument is absent, and that part becomes the pore over here, and that pore is called as the micropyle, and that end is called micropylar end. This much you should know. And then we looked at some food-laden tissue called nucellus. This one. This is of importance to us right now. Alright. In the nucellus, towards the micropylar end, here this side, there are few cells that are going to be differentiated to form MMC. MMC is nothing but megaspore mother cell, similar to our pollen mother cell, similar to that. From there we got the further division, meiotic division, and then we got the microspore, isn't it? Same way here also, from the megaspore mother cell, this cell, where is it? This point, nucellus, from this side of the nucellus is here. You can see nucellus. One cell, one particular cell is going to get differentiated, enlarged, and this cell is our MMC. Further, you know the entire process. MMC undergoes meiotic division. Meiotic division, you get 4 cells. What will be the 4 cells? The 4 cells will be called as the megaspore cells. Megaspore cells. But will all that become megaspore? No. In case of microsporogenesis, I hope you remember, you got 4 microspores. All 4 microspores became pollen. Here it is not like that. Instead, all the 3, among the 4, 3 will disintegrate and only 1 will become the megaspore. That's the only difference here. Everything else remains same. So now we have what? We have 4 megaspores produced. In all the 4 megaspores, we will label as N. Why are we labeling it as N? Because the mother cell was 2N. It underwent meiotic division. Every time meiotic division happens, half the number of chromosomes will be present, isn't it? So all of them we are going to label as N. All right. From there, you get one of them, meaning there were four of them, three disintegrated, you got one megaspore N. Right. This one cell having one nucleus is going to undergo a series of changes. How is it going to change? It's going to, inside the cell, only the nucleus will start dividing. The cell remains same. So that is why this is a single cell. This is a single cell right now with inside the whatever is present, that is the nucleus is present, nucleus is dividing. How is it dividing? Mitotically. Therefore, this N will become one N over here and one N over here. This again, this nucleus splits into two. So how many mitotic divisions it will undergo? 3 mitotic divisions it will undergo. And you get 8 nuclease now. 8. Alright, 8 nucleated structure it is right now. Again, right, but it's only one cell. Inside one cell, you have 8 nucleus present. These 8 nucleus are going to rearrange itself. That's what is going to happen. All right. So how is it going to rearrange? It's going to rearrange in such a way that 3 among them, let's say this 3, will go towards this end. Which end is it? It's called as the chalacel end. What is chalacel end? The going back to the structure of ovule, remember the place where micropyle is present, we call that as micropylar end or micropylar pole. The opposite end is chalacel end. So what you are seeing here, the ovule and the embryo sac is drawn like this. Here it is ulta, upside down structure you are seeing. Please keep that in mind. So you have 3 nucleus on this side and 3 nucleus on this side. How is it? Like this. Alright, 3 over here. Each nucleus will start getting a plasma membrane around it. Therefore, all these nucleus will become cells. So, these 3 nucleus became 3 cells right now in one corner. Other corner again 3 cells because all the 3 nucleus got plasma membrane around it. Now, 2 more nucleus are left that will go and wait in the center. And that will have only one membrane around it. Therefore, what does that mean? A 2 nucleated structure is in the center having a single plasma membrane around it. So, one nucleus, another nucleus. So, how many cells we have right now? 3 plus 3 plus 1. That is why you have 7 cells but 8 nucleus. So, this is your question. Okay, 3-mark question. Why is the embryo sac having 8 nucleus but 7 cells? Explain this. Simple. From megaspore you're taking, explain there are 3 mitotic divisions happening resulting in 8 nucleus. 8 nucleus will rearrange and then how is it rearranging? 3 in the 3 in the chalacel area, 3 in the micropylar area, 2 in the center. But the formation of plasma membrane doesn't remain the same. Only for the whatever is there in the poles will get plasma membrane around it. Therefore, 3 cells here, 3 cells here, 1 in the center. That one cell will have 2 nucleus. That much you're explaining. Now have a look at the structure. Embryo sac. What are the names of these cells formed? Also, you should know. The cells in the chalacel area is called antipodal cells. The ones that are there in the micropylar end is called as synergids and egg. Whatever is on the side, we call that as synergid. Whatever is in the center, we call that as egg. In the center is very easy, central cell with 2 polar nuclei. This much you are writing, you are getting 5 marks. 3 to 5 mark question it comes, you are able to answer. I'll give you more PYQs, previous year questions in the end of the chapter. Please continue watching. Thank you.